Good morning, good afternoon, good evening. Welcome to the International Symposium of Cardiovascular Genetics. It is a pleasure to have you with us in this, I think, very interesting event. In the next 2 days, and then in a week from today, we'll have different lectures from different parts of the world talking about very important topics in cardiovascular genetics. Today, we are, it's 6 o'clock in Brazil, p.m., and I know that there are people from all over the world with us. Unfortunately, Dr. Ernst Hofs will be not available tonight with us. He had a problem, and he asked me to let you know that he will not be participating in the discussions during the event, this first part of the event. I would like to thank the lecturers to be with us tonight and in the other days, and I will start presenting our first lecturer. She is Dr. Ida Vanessa Dodderlein Schwartz from Universidade Federal do Rio Grande do Sul. She's an associate professor of the Department of Genetics at our university, Universidade Federal do Rio Grande do Sul. Professor at the graduate courses in genetics and molecular biology and medical science at URGS, and leader of the research group evaluation of health technologies for clinical genetics. Professor Ida will be talking about basic concepts in genetics. It's my pleasure to have you with us tonight, Professor Ida, tonight here in Brazil.
Yes.
And
thank you.
More comfortable to be with us.
Okay. So, hello to everyone who is attending the meeting. It's a real pleasure for me to participate in this symposium. First of all, I would like to thank Professor Stein for inviting me to participate. I will talk to you in the next 30 minutes about basic concepts on genetics. It's a difficult task because 30 minutes is not enough. We have many things to share with you, but I select some topics that I think are important. And first of all, I will try to put my slides on the... Is it okay or not?
Yeah, it's okay.
Oh, good. So, thank you again, Ricardo, for the nice introduction. And I have no conflict of interest to declare. Here we have the summary of my presentation. I will talk about genes, genome, about the central dogma of molecular biology, some things about epigenetic and gene variants, and I will conclude with some take-home messages. And for the beginning, before we start, I want to introduce you to the father and to the mother of genetics. Gregor Mendel is considered to be the father of genetics. He was born in 1822, so this year we celebrate the 200 years of his birthday. And Rosalind Franklin can be considered the mother of genetics. We can see here the picture that illustrates an edition of the Time magazine. And Rosalind Franklin is very important because she was one of the persons who described the structure of DNA in 1953. So next year we'll celebrate the 70th anniversary of the, not exactly the DNA discovery, but maybe, okay. And we know we are in a symposium on cardiology. We know that the cardiology and the genetics have many things in common. And I want to illustrate this connection, describing to you a patient that we still see in our unit. And I will use this case report to comment on, to you about some genetic terms that we use, like the genetic case. And I think those terms, they are very important that everyone knows, okay? So this is a male patient, an adult patient, and he came to us because he presented very high levels of homocysteine, 431.2 micromole per liter of homocysteine to normal range value until 15. I think every cardiologist knows that high levels of homocysteine are associated with a higher rate of thromboembolic phenomena. And but this patient also presented high levels of methionine and the normal levels of B12 vitamin. This biochemical feature is compatible with the diagnosis of classical homocystinuria. Classical homocystinuria is a genetic disease, and when we think about a genetic disease, this is the first thing that I would like to point out to you. We have to try to classify the genetic disease. Which kind of genetic disease the patient presents? Is a chromosomal disease? Is a monogenic disease? Is a multifactorial disease? Is a mitochondrial disease? Classical homocystinuria is a monogenic disease. What it means? It means that it's caused by the presence of pathogenic variants in only one gene. In this case, the gene which is affected, it's named CBS. This name CBS comes from the name of the enzyme that the gene encodes, the cystathionine beta synthase enzyme. So now we can say that every protein, every enzyme that we have in our body, they are encoded by a specific gene, okay? This enzyme, its function is to convert homocysteine in cysteine. As the patient does not produce the enzyme in normal levels, he presents high levels of homocysteine. The other thing that it's important to point out is that when we are talking about monogenic diseases, we have always to think which is the pattern of inheritance of this disease. In this case, we are talking about an autosomal recessive disease. What does it mean? It's an autosomal disease because the CBS gene is located in chromosome 21, and the chromosome 21 is an autosome. It's not a sexual chromosome. It's not the X or the Y chromosome. And the word recessive means that the individual who is heterozygous for one variant and one pathogenic variant in this gene, this individual is normal. Normal in the sense that he does not present the clinical picture compatible with classical homocystinuria. So the heterozygous for classical homocystinuria, for example, they present normal levels of homocysteine, although they have one variant in one allele, but the other allele is normal. The sequence of the DNA which corresponds to this allele is normal. Anyway, we know that patients with classical homocystinuria, they present two variants, one variant in the copy of the gene which was inherited from the mother, and the other variant in the copy of the gene which was inherited from the father. And they present a high prevalence of thromboembolic events, and so premature death. The IQ can be normal, and the classical homocystinuria is a treatable disease. Although we know that the patient had classical homocystinuria because he had the biochemical findings, as this is a genetic disease, we studied his DNA, we analyzed his DNA to try to find which variants he presented in the CBS gene. What we did? Of course, we did not investigate this patient through the karyotype, because this patient does not present a chromosomal disease, and we use the karyotype to diagnose chromosomal disease such as the Down syndrome. If we did the karyotype for this patient, our results would be normal. So we had to use another method. In this case, we used the Sanger sequencing, and what we did? We analyzed the CBS gene through this technique, and we found that the patient was homozygous for this mutation, okay? Here we have the picture of the Sanger sequencing of this patient, and we can see that in the position 833 of the DNA, he has here a blue peak, which corresponds to cytosine, okay? So this patient, he had two cytosines in this position. This is not the usual base that we found in this position. What we consider as normal for this position is the presence of two Ts. So the patient has a missense mutation where the thymine was changed to the C, to the cytosine. Because of this variant, when we analyze or we predict the effect of the DNA alteration in the protein, what we expect is that in the amino acid 278, we find a threonine. The T here means threonine. And the normal amino acid would be or should be isoleucine, or the I. We know that patients who are homozygous for this variant, this variant is a well-known variant. It is a pathogenic variant. We know that the patients have a milder phenotype and that they are responsive to vitamin B6. So we treated these patients with vitamin B6, pyridoxine, and only using the B6 vitamin, his levels of homocysteine decreased to normal levels. And so we decreased his risk of developing a thromboembolic phenomenon. But this patient was a very special patient, and we know we are also good doctors, and we know that the clinical picture is very important, or are the main things to be analyzed for us to conclude the diagnosis. These patients also presented deafness and hip problems. And we know that deafness and hip problems, they are not explained by the classical homocystinuria. So we thought, oh, this patient should have anything else, not only classical homocystinuria. So what we did, we investigated the patient also through another DNA technique, the exome sequencing. Through this technique, we analyzed all the DNA regions or not all, but most all the DNA regions, which corresponds to the exons of the genes. And we were able to identify two other diseases in the patient. So the patient was found to be homozygous for that mutation in the CBS gene, but he was also found to be homozygous for two other pathogenic mutations. One mutation in a gene which is associated with deafness, so the deafness was explained. It was a genetic deafness, also autosomal recessive, and the hip problems were due to the mutations in another gene, the PRG4 gene, which is associated to a disease called CACP. So now I will point out another basic concept based on the clinical case that I discussed with you. The first thing is that the genetic information in our body, now in the organisms, they depend on two molecules, the DNA molecule and the RNA molecule. The two molecules, they are different. We can see that the DNA usually double stranded, and it has this twist appearance. So we have helix, and the RNA is usually single strand. We know that in the inside part of DNA, we find the base pairs, the bases that are the thymine, the cytosine, the adenine, and the guanine. And I remind you that the guanine always pairs with cytosine and adenine with thymine. Here in the helix, we have the sugars. The sugar of the DNA is called deoxyribose, and the sugar of the RNA is called ribose. They are different, of course. They have different structures. And regarding the bases, we know that they are the same for the RNA and DNA, except from the thymine and uracil, uracil is only found in the RNA and thymine in the DNA. What's important to know? It's important to know that the DNA in our cells, it's located in our nucleus, and it is transformed in RNA, or it produces RNA, it encodes RNA by a process that is named transcription. The whole DNA is transcripted? No, not the whole DNA. The regions of the DNA which correspond to genes are the regions which are transcribed. And when we have the RNA, the RNA goes to the cytoplasm, and in the cytoplasm, the RNA produces or gives the information to the cell to the protein be produced. This process of production of proteins using the RNA as a mode we call translation. We can realize from this picture that we have two products which depend on the sequence of DNA. So the sequence of the RNA depends on the DNA sequence, and the sequence of amino acids of the protein depends on the sequence of the RNA, which depends on the sequence of the DNA. If we have something wrong in the DNA, the RNA will be affected and the protein will be affected. But we have some types of RNA which are not translated. And this is something very interesting and important, because the genes which encodes or produce RNA which are not translated, they are genes involved in the genetic regulation, in the regulation mainly of the transcription, okay? And again, every protein that we have in our organism depends and is produced to those processes, the transcription and the translation. But what is a gene? Gene is the basic unit of heredity. It's a DNA segment that carries the necessary and sufficient information to encode a specific functional product, RNA or protein. And here we have on the top a fragment of DNA which corresponds to a specific gene. We see here a double-strand molecule, and here we have the structure of a specific gene. So one gene is composed by the coding region and also by other regions which are not coded, okay? They sometimes could be transcribed, but not translated. And sometimes they are part of the gene, but they are even not transcribed or transcribed, okay? The coding region are considered to be the most important region of the genes, because most of the disease causing variants are located on those regions. So we have the exons and the introns in the coding region. And in non-coding region, we have the 5' and 3' UTR regions, and also the promoter region, and also some sequences like the enhancers, which are involved in the gene regulation. But what is the genome? We defined before the meaning of the word gene. And when we talk about genome, we are talking about the complete set of genetic information of an organism or cell. And we have the nuclear genome and the mitochondrial genome. We know that the nuclear genome is located in the nucleus, and the mitochondrial genome in the cytoplasm. What's important to know is that the genome is organized in chromosome, right? And if we use the humans as an example, we know that the humans are diploid organisms, so we have always the chromosomes duplicate. I mean, we have one chromosome from which is inherited from the mother, and the another one from the father. So our number of chromosomes is 46. 23 we get from the father and 23 from the mother. And 22 are the autosomals, and we have the pair of the sexual chromosomes. Another thing that's important regarding basic concepts is the words cis and trans. So we can use the chromosome 1 here as an example. We have 2 chromosomes 1. They are called homologous chromosomes because they are almost identical. We cannot see which one came from the father and which one came from the mother, only analyzing the karyotype, at least most times. And when I have 2 variants at the same chromosome, I mean, from the chromosome of the mother, we say that both variants are in cis. But if I have one variant in the chromosome that I inherited from the mother, and a second variant in the chromosome that I inherited from the father, we say that both variants are in trans. Okay. But the chromosomes, they are not only DNA. Actually, chromosomes are made of chromatin, and chromatin is a mix of DNA and protein, the famous histone proteins. We know that the DNA is a very large molecule, and for it to be fit inside the nucleus, it's necessary that it's compacted. And this compactation depends on the histones, because the DNA actually make like some twists. It is twisted around or about the histones, okay? And we know that when we analyze on chromosome, that we have some regions that are more compacted, more condensed, and some others that are not so compacted. They are more open. What does it mean? When we have more open regions in one chromosome, it means that in that region there is more genes, and that those genes, they are active. They are producing protein. Transcription is occurring, okay? And when we have the regions more closed, more compacted, more condensed, it means that maybe we do not have genes in that region, or that those genes are not working. And this is a phenomenon that we call gene expression. So regulation. So we know that the different cells and tissues of an organism, they have all the same genetic information, the genome, but the genome is expressed in a differential manner. What I mean, the neuron is the neuron because in the neuron we have a set of genes which are being transcribed, which are producing proteins. And the red blood cells are red blood cells because in those cells another set of genes are working. So this phenomena of turning on and turning off the genes is called gene expression regulation. And here we have the famous word epigenetics. What is epigenetics? Epigenetics are changes in the DNA that do not involve alterations to the underlying sequence. One of the mechanisms of epigenetics is the histones modifications. So the histones can be acetylated, methylated, phosphorylated, for example. And if this modification opens the structure of the chromosome, what we'll have? We'll have more transcription. We'll have the gene working more. Okay? So this is one way that we explain how the epigenetics acts in our organism. And about our genome. We know that most of our genome is made up of non-coding DNA. So only 1 to 2% of our genome are genes. And how well do we know our genome? In April this year, in the Science, this paper was published. This is a very important paper because it describes the most complete sequence of a human genome that we have so far. And according to this study, we have in our cells 60,090 genes. Among them, only 19,890 are genes which code for proteins. You see that most genes, they do not correspond to proteins, so they are genes that produce RNA which is not transcribed. So those genes are probably involved in the gene regulation. Now, this is the last topic that I have to discuss with you, the gene variants, because we said that when we talk about epigenetics, we are not talking about changes in our DNA sequence. But when we are talking about gene variants, we are talking about changes in our gene sequence. So this term, genetic variant, is the current term that we use to describe any permanent change in the DNA sequence. This term replaced the term mutation, okay? So variant is a more accurate term because not always variant cause disease. We have the non-causing and the causing disease variants. When a variant presents a frequency greater than or equal to 1% in the population, it's called a genetic polymorphism. So this term polymorphism is related to the frequency of the variant and not to the fact that this variant cause or not cause a disease. And regarding the effect of the variant, we can classify the variant in a benign variant, likely benign. We have those variants, the VUS, that we do not know exactly the significance. We have the likely pathogenic and the pathogenic variants. But we also have another ways to classify a variant. We have also the somatic and the germline variants. The somatic variants are not transmitted to the next generation because they occur in any cell division from the first cleavage of the fertilized egg. So those variants are not inherited. And we have the germline variants. Here we have the illustration of germline variants. Those variants, they are inherited, and if they are inherited, the individual has the variant in all cells. Another way to classify the genetic variants is in chromosomal or gene variants. When we are talking about chromosomal variants, we are talking about variants that affect the number or the structure of the chromosomes. So when we have a aneuploidies, we can have one chromosome in excess, or like in the case of Down syndrome, or one chromosome can be missing, like in the Turner syndrome. And when we are talking about gene variants, we are talking about the changes in the DNA sequence that can involve from a single nucleotide to a few base pairs. Chromosomal variants can be detected by karyotype or by another genetic method, such as the chromosomal arrays. And the gene variants, they are detected by methods such as the Sanger sequencing, exome sequencing, genome sequencing. And the gene variants can also be classified in synonymous, missense, nonsense, and splice site variants, for instance. We have the synonyms variants when the nucleotide changes without changing the amino acid. So here we have the C, and it was changed to A. Even so, the amino acid is arginine. So we do not expect that this variant is a disease causing. When we have a missense mutation, we have changing the amino acid. So we have here the G was changed to a C, so the arginine was changed to proline. And when we have a nonsense mutation, the change in the nucleotide generates a stop codon. Here we have the G being replaced by the uracil and generating a stop codon mutation. The splicing site variants, they are located usually in the beginning or in the regions near to the beginning or to the end of the exons. and they affect the splice of the introns. And we can also have the frame shift variants, where we can have the insertion or deletion involving a number of base pairs that's not a multiple of 3, which disrupts the throughput reading frame of a DNA sequencing. So here we have an example. We had the insertion of an adenine here before the CGC, and we see that we have not only one amino acid which is altered, but many others from this one here. So to finish, I have my take-home messages to you. The first one is that we have come a long way since the discovery in 1953 of the double helix, the twisted ladder structure of the DNA molecule. And as our knowledge advances, the number of factors associated with each system and process increases. Thus, even for single gene condition, the monogenic diseases, the genome, the environment, the epigenetic factors must always be taken into account. And then nowadays, technological improvements are not as relevant as our efforts to analyze the exponential amount of data that we are capable of generating. So thank you very much again for the kind invitation for participating in the meeting, and now I'm available for answer the questions.
Professor Ida, thank you very, very much for your lecture. It was brilliant, and I think our symposium start with the right fit. We have some questions, and I will ask you to follow me in short questions and quick answers, okay?
Okay.
Can be?
Yes.
I will ask you a question from my side, my own. In one of your slides, you show the expression monogenic and the other expression polygenic. Yeah. I know this is very basic, but we are talking about basic concepts. Can you extrapolate a little bit about this? Because nowadays in cardiology, we have the polygenic scores. What is monogenic and what is polygenic? Can you explain to us?
Yes, this is something very important, Ricardo. As I told you, that we do not have time for a longer discussion, but when we talk about monogenic things, we are talking about usually diseases, but not only diseases. We can have conditions that are monogenic, which depends the expression depends of only one gene. This is monogenic. Polygenic is different. Okay, I prefer the term multifactorial, not polygenic. Okay, but there is some discussion about the use of both terms. But in general, what the term means is that the phenotype depends on many genes to occur, and of course, those genes they have to have variants. But the variants, they are variants of small effects. Okay, so this is the difference.
I have a question here from Mayo Clinic, Dr. Felipe Vieira. Relevant for cardiologists, why there are some relatives who inherit a pathogenic variant and do not express the disease? Yes, that maybe sometimes you'll see twins, and one will die from the disease with 13, and the other will live up to 90 and die with prostate cancer. It's a very good question.
Okay, but the disease that you talked about prostate cancer, right? You not Filippo. Yes.
No, the
Yeah,
question
no,
from
I know,
Felipe,
no, just for you, because cancer in general, I answer to
it
Filippo.
was an example.
No, okay.
Forget.
Because cancer in general, it's a multifactor or polygenic disease. Okay,
Perfect.
when we are talking about cardiological conditions, okay, I don't have the frequency, but most conditions they are autosomal dominant. Okay, when we are talking about autosomal recessive disease, in general, we expect that the siblings, the brothers and the sisters, they have a phenotype very similar. Okay, of course, they can be different, but when we are talking about autosomal dominant diseases, it's not like that. Okay, we can have a higher grade of different expression, and this we call this like a different expressivity, and it can depend on other genes, okay, and also on other influences like the environment.
Okay.
Actually, we do not know exactly, okay, and the linear the cutoff for us to say that one disease is monogenic or polygenic is not well established.
I have a question from Mary Stewart from Melbourne, Australia.
Yeah.
She's in tomorrow. It's tomorrow over there. Hello. Thank you very much for your question. It's a simple question. Maybe you can explain to us what is a haplotype?
Okay, haplotype. This is a good question.
Thanks, Mary.
Okay, we called an haplotype when we have certain variants at the same chromosome, okay, that they are always together. They are in cis, okay, so it's in the same chromosome, and they are transmitted together, okay. So we can have a variant A in some gene, and this variant always occurs with the variant B in the gene C and with the variant D in gene E. And they are always, those genes are in the same chromosome. This is the variant.
Thanks.
The haplotype.
I'm very happy that we have people from all over the world, Melbourne,
I'm
Australia.
very happy too.
I am proud, and I'm very proud that to invite you, because there is something that caught my attention. If you gave this lecture as a class almost 50 years ago, maybe I will not be a cardiologist. I would be a geneticist, because it was gorgeous and it was very, very clear. But I have another very simple question. This one is from myself again, Porto Alegre, Brazil. You showed us something about exons and introns.
Yeah.
So the non-coding part of the DNA, the gene, is an intron, correct?
The coding part of the gene is exon.
Yes.
Okay, but the non-coding part of the gene is not only the introns. You know, the coding is the exons, and the no-coding is the introns, but not only the introns.
Look, my question goes in this direction. How dark, inverted commas, are the introns?
How dark in what sense?
In sense of we are studying genes and variants in the exons and at the splicing zone. What happened in the introns?
Yeah. When I don't know if I understood your questions very well, okay? But we know that we have some diseases which are associated with a high frequency or at least we have with some variants located in introns, okay? And not only in regions near to the exons. Sometimes we have deep intronic mutations that we do not see those variants if we use techniques such as the exome sequencing.
Okay, because in cardiology, usually in cardiology, in terms of these monogenic diseases, pure monogenic diseases, let's say, we are studying the splicing zone and the exons with the panels.
Yeah.
We do not use exons and whole genome sequence as you use in genetic. This is why I called inverted commas dark side of the gene, because we are not very used. And I think that, for example, a disease as Brugada, Dr. Luciana Sacilotto will talk about this next Saturday. Brugada, even with an incredible expression in the EKG, only 30% of the individuals will have a positive test. So where are the problem? And I think that we will need to study, at least in cardiology, a little bit more the intron. This is why I was with this thought. And you probably study another kind of diseases that are much more related with this part of the gene.
Yes, so two things in this regards. The first thing is that, of course, some mutations can be located in the introns, and if we do not search for them, we will not see them. So we need better techniques. In this case, the genome sequencing. We have another techniques, okay? We can you analyze the mRNA, RNA, okay? We can do transcriptomes analysis. But if we are thinking on DNA, we can use the genome analysis, the genome sequencing, but the variant not always is in the intron. It can be located in the promoter region as well. So we can, but the second thing is, Ricardo, is that we still have some genes to be discovered, you know. We can have some patients that did not find anything even in the genome sequencing, and they still can have something that unknown. Okay.
For sure. Okay. I would like to thank you very much for your participation. It was gorgeous. This is a very informal environment because we are friends and colleagues. This is why we have this kind of communication. Maybe people who does not know us may think they are very informal, but we we like science very much. And Professor Ida gave amazing class in terms of basic concepts in genetics. In 1 or 2 minutes, we'll come again with Professor Filippo Vairo from Mayo Clinic. And then before Filippo Vairo give us his talk, I will show you something very, very, very nice for us. Okay? Bye-bye, Ida. Thank you very much.
Bye.
It was great.
Thank you.
Pronto. Hello, we are here again for the second lecture of our International Symposium of Cardiovascular Genetics. And before introducing Dr. Felipe Vairo, I am very, very happy to say that this symposium is also a discipline of our post-graduation course in cardiology and cardiovascular science of Universidade Federal do Rio Grande do Sul. And at this international symposium, we have 50 students who subscribe to be with us during the symposium. And it was very, very interesting that 14 of these 50 students, 14 are from other cities and other universities than our university. So there are people from Ceará, Buenos Aires, Niterói, São Paulo, Santa Maria, and other cities from Brazil and Argentina. So the next topic that Dr. Felipe Vairo, who is a medical geneticist and a medical director of the program of rare and undiagnosed disease in the Center for Individualized Medicine. He is an associate director, translational omics program, and associate professor of medical genomics, all of these titles from Mayo Clinic. I remember when Filippo and I were talking in 2016, when I returned from Spain, and now he's a very big man. Remember that moment in Zona 18, Felipe? Yeah. So Filippo his talk will last 40 minutes because he will divide the talk in two. The first part will deal with sequences technologies, and the second, variant interpretation. Am I right, Filippo?
Correct, yeah.
Yes? So I'm very happy to have you with us, Filippo, and be my guest to start your talk.
Thank you very much, Ricardo. It was really nice to be invited, and I agree with you. The topics are amazing here, and I think we have to discuss this type of things since like the basics of genetics, and Professor Schwartz gave a wonderful talk and made my life way easier, and they're gonna see why. And thank you for that, Professor Schwartz. And then I'm trying to come up with some topics actually that are more practical than only theoretical. I think we have to discuss these things because the cardiologists, they have been ordering more and more genetic testing. So could you please confirm if you can see my slides in presentation mode?
Yes. Awesome. Go ahead.
Great. So before even I start talking about the sequencing technologies or the variant interpretation, I think I have these general comments so we can start. And the first thing is, regardless of the type of testing that you order, we have to trust the quality of the laboratory or which technology they are using, and if you trust whoever is interpreting the sequencing and the variant results, right? So I think that's the main message here. Regardless if you're going to do even whole genome sequencing, and we are going to discuss this term in a moment, but someone says, okay, I'm going to do like the most expensive tests ever. If we don't have good people analyze the data, it means nothing, right? We can spend a lot of money, and probably it's going to be useless for your patient. Another thing is we have to understand the test limitations, if it's Sanger sequencing, panel, exome, genome. And then we have to evaluate the cost and time effectiveness of the type of testing that we are going to do. For sure, if you need some results quick because the patient is the transplant list or because it's in ICU, of course, we cannot rely on doing a more comprehensive test because it's going to take more time than doing more targeted tests, right? And then for sure, we have to understand the results interpretation. So when we talk about types of variants, if a variant is likely benign or VUS, a variant of uncertain significance, I think that's the most important thing, and we are going to discuss this in a moment. So I'm assuming that you have always seen this slide here, and everything changed in our field, or the field of genetic testing, around 2007, 2008, when the sequencing costs dropped very, very high, very fast. And why is that? It's because we moved from what we call the first generation sequencing, and here I'm showing you a timeline. So we started doing first generation sequencing, which would be like the Sanger sequencing that Dr. Schwartz talked about. And then we start doing what we call the second generation sequencing, which is our NGS, next generation sequencing. And then nowadays, we are in this era here using third generation sequencing now. And the difference between second generation and third generation is the type of reads that we are getting from the sequencing machines. We are going to talk about this in a moment. Some definitions that I would like to highlight here is next generation sequencing is not a test. Next generation sequencing is a technology. There are multiple tests that can use next generation sequencing. So whatever you are discussing with your patient or your colleagues and say, oh, let's do a next generation sequencing, this doesn't mean much. We have to understand if you're going to do a multi-gene panel based on NGS, or you're going to do an exome sequencing or a genome sequencing based on NGS. So next generation sequencing is the term for a technology. And then we have the exome sequencing and genome sequencing. And Dr. Schwartz talked a little bit about exome sequencing, meaning that we sequence the coding parts of all the genes at the same time. And then genome sequencing would be the coding plus the non-coding parts at the same time. But what we have to realize here is that there is no such a thing as whole. Okay, so there are many gaps due to sequencing artifacts and due to the type of rearrangements that we have in our DNA, which means that there are many regions in our genes that we cannot actually sequence. So there is no such a thing as whole exome or whole genome sequencing. We are trying to eliminate this word from now on, okay? Professor Schwartz talked about the gene structure, and just before I talked about the sequencing technologies, I want to highlight it again. So when we say that we are going to sequence a gene, usually what this means is that we are sequencing the green parts here, which are the exons, right? And then we have the pink parts here that are the introns. And then because of that question about if the introns are the non-coding parts, yes, they are non-coding parts, but you can see that in the gene, we have multiple other parts that are non-coding as well, which can be related to disease in the future when we start doing more discovery, right? So when we do a gene sequencing, now means that we are sequencing the gray parts, the green parts, and part of the green, the boundary between green and pink here, okay? So talking about Sanger sequencing, what this was one of the first technologies that we used to sequence our genes. And what does it mean is that we have those base pairs. We go to the lab, we connect these base pairs with some sort of a reagent which lights them up when we use a special camera, and then the sequencer actually is like a very special camera to capture all these lights that come from these base pairs. And what happens is that each cycle of the sequencing, and then I don't want to go into like the PCR and other technologies, but what I mean here is that each cycle, when the correct base pair aligns to the reference that we are talking about, it stops, right? So there are some base pairs which have these ligands that make the sequencing reaction stopped. So for example, the cycle one, when the A that is the correct base pair aligned or ligates or aligns to the right region, it stops. And then we come with the second cycle, and then the second cycle it stops when the correct base pair aligns, and then cycle 3 and so on. So you can see that there are multiple cycles, and we are sequencing the same strand or the same sequence of a DNA, one base pair at a time, right? So usually each read is around 300 to 1,000 base pairs, but we have some limited quality in the first of these base pairs because of some primers and so on. And then the sequencing quality, it's suboptimal going to the end of the read. So with Sanger sequencing, we have very good reads between, let's say, 50 base pairs up to 800 base pairs. So anything lower than that in that read, and then beyond that, it's a little bit lower quality. And then the Sanger sequencing has a limited utility for what we call the low-level mosaicism. Mosaicism means that there are different types of cells in our body with different types of variants, right? So Sanger sequencing, because of the limitation of the sequencing machine, or the sequencing reagents and the technology, when you have cells that have a group of cells that's below, let's say, 15 to 20% of the cells, the Sanger sequence is not the best approach to capture those mosaic cells or chimera or whatever we want to call them. This is not super important for cardiology, but for some other diseases, this is very important for cancer, for example, and for others. So why I'm talking about Sanger sequencing? Because we are gonna discuss what we have been using now more in cardiology, right? Which is the MGS, or the massive parallel sequencing. So as I said, Sanger, we do base pairs by base pairs, and we are gonna sequence the whole DNA strand here as I said, one base pair to the other. With the MGS, or the massive parallel sequencing, we chop this DNA. So we cut them in multiple parts, and then we sequence each of those pieces of DNA at the same time. So we save a lot of time and a lot of money doing that. And there are many types of machines, and this is just one of the most common that sell by Illumina, which is but there are other companies as well. But so how we do the next generation sequencing? So we have the genomic DNA. We fragmented this genomic DNA, have multiple pieces of this DNA, and then we use some adapters, and then we have many reagents, and then we throw all these multiple pieces of DNA in a flow cell, which has other ligands here that's gonna connect to the adapters that we put, or we added to our genome DNA. And then based on multiple cycles here, we have what we call these bridge amplification cycles. And you can see that we have clusters here. So each cluster here, it's from the same read. So we're gonna have multiple reads for that specific position. So you can see here that we have, again, a camera. So that's a fancier camera than the camera that we had in the Sanger sequencing machine. Now we have this NGS machine. So at the same time, instead of capturing base pair by base pair, this camera captures multiple base pairs at the same time, based, again, on lights. And each base pair has one light, and this camera captures those at the same time. And then after all this happened, we have to have powerful computers to actually connect all these pieces together in the right place, so we can call the variants, right? And that's what we see. All these reads here. So you see these adapters here in red, and these in blue are the DNA, the reads that came from the sequencer. And then we have multiple reads that we use some computers to align to this reference genome here. And then, for example, in this region here, we have all these reads, not this one because of the adapter, but all these reads here that would have good quality to call variants in that specific region, and so on. So each read here would refer to this specific piece of DNA in the reference genome. And that's how we see this in the computer. Instead of having one letter by one letter, as in Sanger, we have multiple letters at the same position. So if we have multiple letters who are calling the same read there, or the same variant there, it means that it's a high quality, or you are more confident in that call because we have multiple reads. And we're going to talk about coverage and that in a moment here. So when I'm talking about here exon capture, it's the same principle when we are doing multi-gene panels, because, as I said, usually we sequence the exons of the genes, right? So these are the steps that needed to happen. So you have exome or a multi-gene panel sequencing. So as I said, we first cut the DNA in multiple pieces. Then we use some reagents and some baits to actually select the regions that we are interested in, because remember, the DNA has a lot of things, not only exons, but then we have these baits here to select exactly the exons that you are interested in. And then we actually, after all these steps here in the lab, we can select the regions that we are more interested in based on some magnetic, or depending on the technology, something different with other reagents. But the thing is, here in the beginning of the sequencing, or even before going to the sequencer, we have multiple steps that the lab has to do, so we can select the regions that we want to sequence. Okay, and when I say that we want to sequence, that's what we see in a computer. We cannot make sense of it for sure. So here there are a lot of quality control, numbers and letters and so on. So we need some powerful computers to actually get this information here and transform this in actually a gene sequence. And here it's this gene here, ARPM2, that reference. So the computer connects all these sequences here that they found in multiple reads, put them together, and then we go and analyze and get the sample here and align to the reference genome. That's what we usually see when we are looking at the data. And now we start discussing what I told you about coverage and depth, right? Many times we use those terms interchangeably, and they are not, they are different. So when we talk about coverage, it's something horizontal. Coverage means that, and here I'm gonna show you this blue squares and rectangles here are the exons of a gene. Doesn't matter which gene, right? So when I say coverage, means that those exons have some sort of reads that covered those regions. And you can see that there are regions here with this amount of reads, and there are regions with this amount of reads, right? So I would say that all these exons have some sort of coverage, but then when I'm talking about depth, means that the number of reads that go in each region here, right? So when we are doing multi-gene panels, when we say, oh, let's that region has 150x, for example, this means that that region is covered, but the depth of coverage is 150. Means that there's 150 reads at that same position, so you can confidently call that variant because 150 reads call that variant that is a T instead of an A, for example. Okay, so keep this in mind when we use the word coverage in Portuguese or depending on the lab. Probably what they mean is the depth. It means that how many reads we have in that region. Okay, so now we are talking about the variant calling, and here, as I said, all these letters, because we have this depth of reads like 3, 6, 9, 11. So, for example, in this region we have 11x of depth. And then in this region here, we see that this would be like the reference. This is the reference genome. So, for example, the reference would be a T, but this patient has, or this individual has 4 T's and 5 A's, meaning that this patient is heterozygous for an A in that position. Okay? So the next one here is this one. The reference would be an A, but then actually our patient here has only G's, meaning that this patient, he is homozygous for an alteration in that position. Okay, and this patient here, for example, has the C would be the reference, but then there are multiple C's, or most of the reads are C's, but there are 3 here that are T's. So what this means is that this is a low frequency T, probably related to a mosaicism, for example. Okay. And then the last one, it's a lower coverage region. So we have only 5x here. And one of these, instead of an A, it's a C. So probably we are dealing here with an artifact. And that's why depth of coverage is very important. We need to have multiple reads at the same position, because if one variant there or one read shows you a variant that is not the reference, we have to be confident in calling this as an artifact instead of maybe, for example, let's say a mosaicism, or depending, if you had like only 2x here, an A and a C, how could you possibly tell if this is an artifact if this is 50% of all the reads that you have? Maybe you had the heterozygous, right? So that's the importance of knowing the depth of coverage for that specific region. And another thing that's important about the depth of coverage is when we have multiple reads, and then multiple can be, depending on the lab, whatever from 50x to 1000x, let's say. Because you have multiple reads, and multiple cases at the same time, you can start calling what we call the copy number variants, which are a different type of variants, but they are very important and they can cause disease. So for example, in this region here, since there are no reads, this patient actually had a homozygous deletion. Or for example, if the normal range would be like this region here has less reads, so this patient had a heterozygous deletion, and the same here, if the region, this region we would expect to have all this amount of reads, but we have more than that, then this means that there is a gain of DNA in that region, possibly a duplication. Okay? So to do a quick comparison between panel, exome sequencing, and genome sequencing, and here I want to stop one second because there are multiple labs that they actually sequence the exome, but they sell the panel analysis, okay? And then we have to be aware of that because this type of test would have the same limitations as the exome sequencing, because they're not doing a targeted panel, they are sequencing the whole exome, but they are only analyzing the genes that we are interested in. So anytime that we receive a report, check on the methods and see if the lab did a target panel, a target NGS panel, or if the lab actually sequenced the exome and just analyzed the panel, right? So in this comparison, for sure, gene panels, usually we include less genes than 20,000 genes that we include in an exome sequencing or genome sequencing. The coverage or the depth would be higher in the panels because we have less genes that those reads should cover. The cost, of course, it increases when we are doing a more comprehensive test. And then when we are developing panels, we have to go to the literature and actually decide which genes we are going to include in the panel. And when we are doing exome or genome, it's kind of more agnostic. We are going to sequence all the 20,000 genes at the same time, so we don't need to think a lot which genes we are going to include. But then the analysis is it's more difficult because we are sequencing more, we are getting more variants, and so on. And of course, the counseling efforts as well. So I talked about NGS, but is there a place for Sanger still? And there is. There are some single genes that they are small, or because we have like a biochemical confirmation of that patient. So for example, for cardiology, GLA, it's a gene that causes Fabry disease. So if you have like a biochemical confirmation, your patient has Fabry disease, but you want to know the genotype of the patient, probably you don't need to do a genome sequencing, right? You can select this specific gene and do a more targeted analysis. Sometimes it's going to be Sanger, sometimes it's going to multi-gene panel, depending on the technology, depending on the lab, depending on where you are, right? There are some genes that have limited coverage when we are using the NGS using short read sequencing. So Sanger is useful for those regions as well. Many times we know what the variant is for that specific family, so we don't need to do, again, a panel or a genome or exome sequencing. We can do a Sanger for that specific variant. And then there are some labs that they usually they use Sanger sequencing to validate what they found in the NGS data. So why we would do exome? Because as Professor Schwartz said, we are focusing on the part of the genome that we have more information on. Most of the known disease-causing variants are located on exomes, over 20 or 85% of them, and still it's more cost-effective to do exome than genome sequencing, but this is going to change soon. There are many things that exome miss, so it's important to know the limitations. Even the protein coding genes, we still have some limitations depending on the exomes that we are dealing with, because of the type of base pairs that are in that exome. When we have more GCs, for example, it's difficult to do the PCR, so probably we have lower coverage for those exomes. We don't cover these non-genic control elements. For copy number changes, we can call copy number changes with exomes, but the smaller ones we still miss, again, due to technical problems. Structural changes like inversions or translocations is very hard to capture with exome sequencing. Mitochondrial DNA, we can capture mitochondrial DNA, but exome is not the best or the gold standard test for mitochondrial. And then not super important for cardiology, but for neurology, for example, there are many genes that the common pathogenic mutation or variant is the true nucleotide expansions, and exome sequencing would miss those. So why would do genome? Because we have uniform coverage among all the exons and the introns, because we can call variants in introns, no coding, regulatory, and intergenic. We have a better CNV calling. We can call translocations and inversions, for example, and we have better coverage for mitochondrial DNA as well. Okay. So talking about cardiology, as Professor Ricardo just told us, we are most using panels. We are not using a lot of exome sequencing yet, but we have to be aware of some caveats of doing gene panels. And here I'm just a quick search in Google. You can see there are labs offering cardiomyopathy comprehensive panels, and then this lab calls it like this, and another lab calls similar, and other lab includes arrhythmia, cardiomyopathy panel, and so on. And all these panels, they include different genes. You see hundreds of genes, 200 genes, 100 and something, and 53 genes. So more important than saying that, oh, we did a gene panel for that specific panel, for that specific patient, we need to understand which genes were included in that panel, because if you have a negative result, maybe it's because the variant that is causative for your patient's disease was not included in that gene panel, for example. And actually, there are a lot of discussion on which genes should be included in the panels, and there are multiple other publications. And for example, this group for dilated cardiomyopathy, they evaluated 51 genes that are included in most of the gene panels that are sold by the clinical labs. And actually, what they found is that only, let's say, 19 of those 51 genes have at least moderate evidence of being related to dilated cardiomyopathy. And the same for hypertrophic. So out of 57 genes, even less than 11 genes had moderate or at least moderate evidence of being related to hypertrophic cardiomyopathy. So who decides which genes would be included in those panels, right? So there is a group that's called the ClinGen group, and they are from a group from the American College of Medical Genetics and Genomics. So they put together expert panels in multiple diseases, and there is a group focusing on cardiovascular genes. And you can see the list of phenotypes that they have been working on, and actually they decided which genes actually have the limited or moderate or high evidence, strong evidence of being related to those phenotypes here. So it is freely available. You can go to the website and see exactly what genes they suggest or recommend to include in those in panels for those specific conditions. So regardless the type of testing you did, you have to find the causative variants for your patient, right? And how we approach that, when you do multi-gene panels, you're going to see thousands of variants, right? And we have to filter these variants and prioritize what we feel is they are the most important variants for our patient. And how we do that, we start with thousands of variants. We usually come to like a handful, 5 to 10 variants that we are kind of interested in. And then after finding these 5 to 10, we actually have to interpret these variants and classify them in groups. And we're going to discuss this in a minute. So usually how we filter these variants, it's based on the quality of the sequencing. It's based on where in the genome they are, how frequent they are in the sequencing data, how frequent they are in the populations, which type of variant in which gene, phenotype, inheritance, and so on. So we use a lot of criteria to come up with the candidate variants. And then when we come to these variants, then we have to interpret them. And then this is the second part of my talk, actually, but they are connected, of course. So there are multiple groups working on developing guidelines on how we should interpret variants, right? And the most common guideline that we use in the clinical labs is this ACMG and AMP guidelines. And you can see here there are multiple criteria for each variant that a lab is interested in. They have to focus on population data, some in silico predictions, functional data, segregation, and check some databases. And based on the strength of those evidence, they collate this information, and then they assign a classification for that variant. And in this is the most recent guidelines in 2015, and they classify the variants from benign to pathogenic, and with likely benign, likely pathogenic, and VUS in the middle. So how they classify that? The idea is that a variant to be called pathogenic, it means that the variant has a over 99% of probability of being pathogenic. And if a variant has over 90% of being probability of being pathogenic, then it's called likely pathogenic. And the same for the benign and likely benign. But in the middle of 10% to 90% of probability of being pathogenic, there is this big group of variants that they are called variants of uncertain significance. And this creates a lot of confusion. I know it's not only the cardiology space, it causes confusion in any space that we are doing genetic testing. The thing is, in that specific 2015 guidelines, actually, they say that a VUS should not be used in clinical decision making, which is very bold to say that, because we know that there are many VUSs in this space here that they are more likely to be somewhat or to going towards likely pathogenic than going to likely benign, right? So if a variant has, let's say, 89% of chance of being pathogenic, they are very much, they are different than a variant that has like 11% of probability of being pathogenic, right? So we have to keep this in mind. So when we say that a patient has a VUS, it's not just a VUS. It's not benign. It's a VUS. It could be even like more towards like likely pathogenic. So we have to be cautious when we are talking about this type of classification here, okay? So there is a group in UK actually that they classified the VUSs in different categories as well. So they use this terminology here, the variant that's close to likely benign, it's ice cold, and a variant that is close to likely pathogenic, it's hot, based on those criteria that I told you about, the number of criteria that probably they still cannot be classified as likely pathogenic, but they are close to it. And then we have this criteria in the middle of those. So the American College doesn't use this. They put everything in the same bucket as a VUS, but the UK group, for example, they use this terminology as well. So those guidelines, they come up on in 2015, and right after them, they started coming with some updates, because we are developing a guideline that in theory should work for 20,000 genes, and we know that this is impossible, right? So for each of those criteria now, there is a group focusing on recommending some adaptations of those criteria. So since 2015, we now have at least all these general updates. So when we are talking about, oh, let's use the most updated guidelines for variant interpretation, it's not the 2015 anymore. It's the 2015 plus all these updates that they are coming year after year here. Okay, so be aware of these changes. And then not only that, there are some specific guidelines for specific conditions. And for the cardiology, for example, I'm just bringing a couple here. There is a group that they publish the specific criteria that should be used for LDLR, specific criteria for genes that cause dilated cardiomyopathy, and just one example here for hypertrophic cardiomyopathy associated with MYH7, there is a specific criteria that should be addressed. So forget about the 2015 guidelines, and then when you're dealing with MYH7 variants, you should follow these criteria here. And you can see that there are some criteria that they recommend that we cannot even use for this specific gene. For example, those in light gray here. Okay, I don't want to go over all of those because in a matter of time. But then, so they publish this specific classification for MYH7 as a phase 1, but they are in the works of the phase 2, creating specific criteria for all the other genes here that cause hypertrophic cardiomyopathy, dilated or restricted, for example. Okay, so keep this in mind. Two of those criteria I want to touch on because I think that's very important for us clinicians, right? One of them is this one. So anytime that we say that a variant is rare, we think that this is important. And after sequencing thousands or millions of people, actually, we know now that 54% of variants are only seen once in all those millions of patients. Okay, let's say not millions, here it's hundreds of thousands of patients. So 54% of the variants are actually rare, only seen once in the whole database of the population database, right? So actually rarity is actually common, and we usually put a lot of strength in the variants being rare, but I just want to show you that now the guidelines actually, they use this criteria of population database or variant being rare or not seen in population database only as a supporting, like gives a single point only, which probably is not going to change the variant classification at all. Okay. Another one that's important for cardiology is there is one classification or one criteria for de novo variants. In genetics, when we see a variant that appears de novo, meaning that it's not inherited from the parents, we think that this is very important. And we should, because it's novel, right, or appears de novo, sometimes novel. But for cardiology, there is a specific, so the new guideline actually for cardiology, they say that if a phenotype is consistent with that gene, but not highly specific, or there is high genetic heterogeneity, which is true for cardiology, for cardiomyopathy, for example, because there are multiple genes that cause a similar phenotype. Actually, a variant being de novo in a single patient gives us only 0.5 points, which would be supporting again, which is just, again, 1 point in the whole list of our criteria. Probably it's not going to change our variant classification at all. So keep this in mind when we are dealing with de novo variants or rare variants. Maybe this is not sufficient to call a variant as being causative for a patient's phenotype. So what we can do as clinicians to help resolve these uncertainties. And here I'm bringing again all the criteria that usually the clinical labs use. And I can show you all these highlighted in red here could be applied if we, in our clinics, we do something else to help the labs to reclassify the variants. For example, knowing that the specific variant has been seen in other people with the same phenotype, if we can segregate this variant in other affected members of that family, if we confirm if we test parents and confirm that the variant is de novo, it's rare in cardiology, but there are some recessive genes, if the variants are actually in trans, or if we provide more information on phenotype for the lab to reclassify the variants. All these criteria here can come from supporting to very strong, depending on how much evidence we provide to the clinical lab. So we as clinicians, we can help reclassifying variants, testing affected individuals, connecting with other providers, for example, or checking some databases. And I'm just putting this out there here. There are population databases, there are disease databases, there is CleanGen, which provides some variants there in their website. But actually there is a lot of groups working on these undiagnosed cases, so we can connect with those groups and share our information, the information of our patients, and probably finding other patients out there with the same variant. And then when we combine these efforts, we actually can reclassify these variants. So keep this in mind, that we can help reclassify variants. Another thing that I want to highlight here is in silico predictors. We put a lot of strength in silico, and we shouldn't. And this is one example here for BRCA1 and BRCA2, and I'm going to show it in a minute for cardiology. So the two most common predictors that we use, they are only accurate for BRCA1 that would cause like breast cancer, and we know that how important it is. They are only accurate in 60% of the cases. And we keep using them and saying that we are going to classify a variant based on whatever these in silico tools they say, right? And so the accuracy goes below 60%. So for cardiology now, a group put together a lot of effort in analyzing multiple in silico predictors to help in interpreted cardiac conditions. And they come up with what they call this cardio boost, which outperforms any other in silico tool, but as you can see, for cardiomyopathies, it's only good 63% of the time. It's better than anything else, but still, if we put a lot of strength in silico tool, we are going to miss or we are going to call wrongly variants 40% of the time. For arrhythmias, it's a little bit better, but still, it's not perfect at all. So again, de novo variants are not pathogenic. Rare variants are not pathogenic, and if the in silico tools call a variant deleterious or pathogenic, we still have to be cautious about this information. Okay, a couple of words on reanalysis. So for undiagnosed diseases, for Mendelian diseases, we know that analyzing this data over a course of 1 or 2 years increase the diagnosis rate in over 10% of cases. In 5 years, increase up to 20% of cases. And actually for cardiology, this group in the Netherlands, they in 4 years, they reanalyzed some variants and they reclassified 15% of those variants, Most of them, they were downgraded. All these VUSs were downgraded to likely benign. So it's important as well for our patients to reduce this uncertainty. But then some of those variants, a minority of them, but some of them were reclassified as likely pathogenic. So we have to be aware of that. We don't need to do more genetic testing. We just need to reevaluate those variants in a couple of years to see if more information is already out there that could help reclassifying variants. One thing that we did here, not for cardiology, but for nephrology, and I think that could be applied to any specific phenotype, right? We put together a group of geneticists, experts in that disease, pathologists, researchers, everybody together. We discuss these results together, and we provide this follow-up to our patients. And that's what increases the likelihood of calling VUSs as a variant that maybe it's a hot VUS, maybe it's important, right? Because we have this multidisciplinary group, and I think for cardiology, we should do the same. So my final remarks, regardless the type of genetic testing, it should be tailored according to the comfort of the provider with technology, how we would interpret the results, depending on the patient's phenotype, and then urgency of this test. The diagnostic yield of panel, exome sequencing, or genome sequencing depending specifically on the phenotype that we are dealing with. So maybe for cardiology, we don't need to do exome sequencing. Maybe having a curated gene panel would suffice, and then doing genome sequencing to search for intrinsic variants, for example, or non-coding variants would be the next step. Genomic data interpretations optimally performed when we do this in a multidisciplinary team. And remember that reanalysis of data can help resolve some cases. And I'd like to acknowledge all this group here because I don't work alone, and I'm happy to take any questions that you might have. affected as a proband because the variant was mosaic in the parent, so not all the variants were there. It was germline in the proband, but then we would call it de novo because the parents they didn't have the phenotype. But then when you go back and do next generation sequencing, we see that one of those parents they did have some cells that had the variant. So this is what I mean with mosaicism. But to answer Fernando's question, we can do this type of testing, multi-gene panels, exome sequencing or genome sequencing. If you extract the DNA from the heart, you can search for variants that happen only in the heart. And then probably you can compare to the blood and see if they are somatic or not.
I'm sorry to say that there was a cut in the YouTube transmission, Filippo, and some questions were not transmitted. But I will ask you something. Marcelo Bittencourt is asking, for those who works with large panels, you are in favor or report in VUS from genes that are of limited evidence by ClinGen?
That's the $1 million question. Great question, Marcelo. Thank you for that. I think this would be like personal opinion. It all depends on the type of group that you have to analyze VUSs, right? If you're there by yourself in a small place, you don't want to see a lot of VUSs that you cannot deal with. You don't have the support to do counseling with that family and so on. I would say that it's better to not see as much VUS that you could, for example, in the clinicas where you have like a genetics department who can help with counseling and experts in cardiology. So I don't have like the perfect answer here. I would say that's depending on the how comfortable you are with these results. But there are a lot of people discussing exactly your question about including those genes with limited evidence, right? And then if there is limited evidence, probably every single variant in those genes are going to be classified as VUS because there is for the gene has limited evidence. So for the variant, probably you have even less evidence that it's causative.
Is there any ethic problem in one hand describing this VUS, at the other hand not describe the VUS? What do you think about this?
I don't see this as an ethical problem if in your methods you specify what you did, right? Every lab should put some the fine print there in the lab in the report saying I'm not including VUSs in limited genes or limited evidence genes. Great, you are covered there, right? I don't have any problems with this. And if they included, I don't see as a problem. But then as a provider, you have to be able to counsel your patient accordingly. You have to say, that's what we have as evidence. This variant we don't know yet, and we are going to follow this for some years. Who knows?
Fabiano Poslar, our geneticist here at Hospital de Clínica, he's asking, how can we test relatives for resolving a VUS in conditions with reduced penetrance, like some sarcomeric cardiomyopathies? What would be optimal strategy in this context?
Great question. Thank you, Fabiano. I would say that for genes with reduced penetrance, probably you're not going to reclassify those variants. They're going to keep being a VUS, but as a physician, you will have more evidence to call it like a VUS of interest or whatever you want to name it. Again, I don't think we have to be so attached to labels that the labs call the variant as uncertain significance. We have to come with evidence to discuss with the patient saying, now in your family, 5 of your affected relatives, they have the same variant. Even though the lab cannot reclassify it, I think that's very important for your family. And that's it.
Okay. Dr. Filippo, it was great to have you with us. We are missing you here in Hospital de Clínicas. It was a fantastic talk and very good question from the audience as well. And I invite you to stay with us if you can. And now 1 minute for a glass of water, and then we will return. Okay, soon for the last activity of the night, that is what we call in Portuguese a colloquio. That will be something different probably in this kind of symposium. Thank you very much, Filippo.
Thank you. It was my pleasure. See you.
Ok. Pode desligar. Pode tirar. Hello, we are now in the beginning of the third activity of this evening, and it's my pleasure to introduce Dr. Marcelo Imbronizi Bittencourt, a friend from Rio de Janeiro. Dr. Bittencourt is a cardiologist and PhD in cardiology, a physician at heart failure and cardiomyopathy clinics at HUPE UERJ, Rio de Janeiro, Brazil, and he is the head of cardiogenetics, DASA Genomics. Dr. Marcelo will be a chairman at the interactive roundtable, what we call in Portuguese colloquio, and the title is the Young Doctors and Cardiogenetics. Why young doctors? You'll see very soon. Marcelo, the word is with you. Thank you very much to be with us.
Good evening, Ricardo, and thank you for the invitation. It's a pleasure to conduct this interactive roundtable with these friends, and we will explore very important and interesting topics. But first of all, I would like to introduce our speakers. Here we have Dr. Thais Beuren, cardiologist, PhD student at Universidade Federal do Rio Grande do Sul and Amsterdam University in the Netherlands. Fernando Scolari, a clinical cardiologist and coordinator of the hypertrophic cardiomyopathy clinic and member of the heart failure and transplantation team at Hospital de Clínicas de Porto Alegre, Brazil, fellow in advanced heart failure and transplantation at the University of Toronto, and Fabiano de Oliveira Pozoar, medical geneticist at Hospital de Clínicas de Porto Alegre, PhD in genetics and molecular biology at Universidade Federal do Rio Grande do Sul, Porto Alegre, Brazil. To start, I would like to ask the three of you a question regarding cardiogenetics and the work that you do in your universities. What are the expectations and challenges? And I would like to start with Thais.
Well, good evening to everyone. Thank you, Marcelo, for the question. Thank you, Ricardo, for the invitation also to this event, this lovely event. And well, I think genetics as a discipline, in medical school at least, we don't give a lot of attention, I think. So I think we already graduated as doctors. It's already a challenge regarding to genetics. So I think the challenge starts there, and cardiogenetics even less. So after the residency or during the residency in cardiology, I already noticed there was also a subject that we didn't approach with a lot of frequency. So definitely it's not something that we are that used to to deal with in our tendency. So I think the biggest challenge in Brazil, I cannot say anything else. It's firstly the cost. I think it's hard to how can I say, to spread genetics in general, and especially cardiogenetics, because it has a high cost. So it limits a little bit, well, it limits a lot actually our practice in that sense. Also, even if we have, if you have access to a panel, for example, still I think we have a challenge in the sense that because people don't see, and especially other cardiologists and other doctors, I think we still don't see genetics as an important issue to address. So I think our graduation is really focused on therapy, and we end up by not focusing on family history and seeing more than just one patient that is in front of us. And it's not like we forget, but we don't focus on the fact that the disease has, it came from somewhere, right? The patient is not only the one that is in front of you. So I think because it's not that well spread throughout the doctors, I think it becomes hard also to make people believe it's important, because sometimes genetics doesn't change our therapy and pretty much the therapy, because the prognostic, of course, it changes, but it doesn't change the therapy a lot still. This is also a challenge. So I don't think people focus a lot on genetics. I would say those are the biggest challenges to start with. Not maybe, Fernando will agree with me, maybe talking about the variants, which is really hard to, even if you get a test, people order the panel, still have a lot of challenge on interpreting the panel and really linking the disease to the genetic findings we have.
Okay, thank you. I think it's changing. The panels is becoming less expensive, and to change the therapy, especially in certain high risk groups, we notice that we have changes in these situations too. But Fernando, what do you think? Expectations and challenges.
Hello, Marcelo. Good evening, everyone. First of all, I'd like to thank Ricardo for the kindly invitation for this program and for the introduction. So I think working in an academic hospital, we have several challenges because first of all, we are the center that should be doing this. We are the ones that should be raising the flag and say, well, genetics is important. We should include it on the residency. All the students should have contact with the genetics. And how can we do it on a public health system that doesn't support the cost of genetic testing in the cardiology field? So, well, then we have to be truly Brazilians and find ways to do things, right? So, in our experience, we started with some funding, very little funding with for academic projects that we use it for sequence patients with Sanger. We only had Sanger at the time. Then we got some know-how, and then we got more money just to do some panels for some patients. But that was a very start. And even though you can do only a few patients, maybe that's quite important for that single family. But nowadays, I think because the cost is decreasing a lot in the past decades, I would say when I started working with this, I think a panel would cost like $10,000. Now it's like $1,000. So what is going to happen like in 5 or 10 years? Maybe it's going to be $200 or something. We don't know if the public health system in Brazil is going to incorporate. We know that we have some initiatives to do that. But that shouldn't be an issue for us. We should do at least talking to the patient, say, you have this indication, you should be doing this. The health system doesn't cover. Do you have some money to do it? Because sometimes they do. And sometimes we get surprised with that. And the other thing is sometimes we can do some partnerships with companies. We know that recently there was a large company that wanted to run some tests in Brazil just to have some data, probably. We know, of course, we always have to be with one step behind and say, well, if this is free, maybe I am the product, right? But then there was some of these partnerships of people like giving some panels for us, and some other companies that are interested in finding some diseases, such as like in HCM, on an HCM cohort, probably there are some Fabry diseases patients, and if they are positive, then you need to buy the enzyme. But in the other hand, these companies give you a simple panel that you can use. It's a start, So we need to struggle with these limitations. Of course, we need to understand our scenario, our setting, but we shouldn't be limited by only by the cost. And I think the other struggle is, of course, we are doing these families, we are sequencing these patients, but what should we do with the results? And that's a problem. I mean, when you don't have an experience, or when you're a center that you are alone. And so we have an initiative like with a genetic board. So taking together geneticists, cardiologists, and bioinformatics, discussing together a variety of VUS, and sometimes we changed the VUS. We had an experience with the patients that it was like a VUS, we like to call it a VUS plus, it's not significant, but there was like, we say that there was a nose of a pig, a tail of a pig, and probably that's what there was a pig.
So, a strong VUS.
A strong VUS. So I think taking that together and also opening for other centers, people that have some of these doubts on their sequencing, and bring to our group just like as an open group for discussing. And I think that's very important, because when a center grows, everybody grows together, and more people orders tests, and I mean, the patient's benefits.
Okay. And Fabiano, what do you think about expectations and challenge for the cardiogenetics?
Well,
You are
I
with
have a
Vai assistir depois no vídeo.
Mas
amanhã e depois
I have a different experience here as a geneticist because I'm used to think on rare disease, and so we had this opportunity to investigate many patients with cardiomyopathy for rare conditions like Fabry disease, like mitochondrial conditions, like fatty acid oxidation disorders. But it was very frustrating because we are investigating just the more rare spectrum of possible diagnosis. And when we are talking about cardiogenetics, we start with a transition of genetics to more common diseases. And I think this is a very good transition that we are seeing right now. And I think the next generation sequencing allowed us to have this transition, especially considering some genes like TTN, that's a very large gene. So we now have a more adequate situation to investigate those diagnoses, and I think we have the most important challenge remains to be the funding for the tests. But I think that with the new data, probably we will get to convince the public health system to cover those tests, because can maybe prevent some cases of sudden death, and well, we have some more changes that can provide from doing those tests.
Okay. Now I would like to make some questions according to your experience, beginning with Fernando. Based on your experience with HCM, Fernando, what do you think of the usefulness of cardiogenetics to stratify risk in this condition? Do you think it has a significant role?
Yeah, definitely. I would say that in hypertrophic cardiomyopathy, genetic testing should be just as an EKG. Like, everybody should be assessed. First of all, diagnosis, as well, sometimes we have patients with borderline hypertrophy that you don't know the cause, and it may changes the way you look at the patient. If the patient has that increased septum and has hypertension, and you are in terms of deciding if you're going to put an ICD, let's say, would it change your practice if you know that the patient has a pathogenic mutation? I would definitely would. If the patient is borderline for implanting an ICD and there is a genetic mutation, I would definitely do, because then I know what disease I'm working with. Because in HCM, only 50 to 60% of patients would have a mutation. And sometimes we may be dealing with hypertension-induced hypertrophy. Mm-hmm. So in that singular case, I would definitely change my practice. So yeah, for sure, the presence of a pathogenic mutation can change the outcomes of this patient.
It's the difference pointed by these registries.
Yeah, and
like share.
When you look at the share registry, you know that if you have a SART mutation, you have a worse prognosis than you have if you have a benign mutation or no mutation at all. And then you would have an intermediate outcome if you have a VUS. So because some of those VUSs are pathogenic, actually.
Yeah.
So And if you look on the updated guideline for the European Heart Society for Prevention of Sudden Death, they include that pathogenic mutation could be change your indication for ICD if you don't have other conditions for sudden death.
As a modifier.
As a modifier. So bringing you from a class 3 to a 2B. So I will definitely change practice with that. And something that we were talking before, in the presence of other epigenetic modifiers, the presence of a pathogenic mutation can lead to a worse natural history of HCM. You can have more fibrosis. Sometimes you can have a dilation of the LV in some cases. So understanding what kind of patient you are dealing with changes also the way you're going to follow up that patient. Are you going to follow more closely? Are you going to give like 1 year follow-up, 2 year follow-up, or are you going to see that patient every 3 months, 4 months? So I think that the more we know about these mutations, the more we can change our practice, actually.
Yeah. Thais, you also work with HCM. What's your opinion?
Yeah, I think HCM definitely, well, it needs to be genotype, of course, and not only because of the prognosis of the patient itself, and I always like to highlight that because sometimes it's not going to change a lot our practice in a patient that we already have the disease already established, but it's really important also for the family. So if you have a patient that you have a positive panel or a positive mutation, you can already tell the family or the kids of that patient that they have 50% chance of also having the same mutation and they can also develop the disease. So I think it's really, really relevant and important that you tell a person, well, you might have a chance of having a disease or you do not have the chance of having the disease.
Perfect.
So it's a relief, I think, for people that can have the mutation and they are tested negative. Also, it changes, I think, very important life decisions, even as professional ones, the way you take care of yourself, and even, well, the development of the disease if you actually have the mutation. So yeah, I think it's one of the diseases, well, cardiomyopathy, well, all of them. I cannot choose one more important, I think, but I think it should really be spread around, well, doctors, because I think it's really important.
And Thais, changing a little bit the topic, what can you tell us about your experience working with phospholambda mutation? I know that you worked with this in Amsterdam, and despite it's a hair gene, but it's a very high risk group of gene that we and you have this experience. What can you tell us about this?
Well, so CLN, well, phospholambda gene was actually my first contact with cardiogenetics. And I started working with a specific mutation, which was the R14R deletion of PLN, which also was found to be a founder mutation in the Netherlands. So I kind of started upside down, I think, in genetics, because I started with a very, very specific mutation, and then the people that had the carriers of the mutation, we looked backwards to see which phenotype they developed. So this specific mutation, they could develop, or they can develop ARVC, arrhythmogenic cardiomyopathy, and also dilated cardiomyopathy. Those are the two main ones. Some people, well, don't develop the disease at all, and it's also, it's accordingly to the age, so the penetrance varies with age. So I kind of started looking from the mutation to the phenotype, and this is I kind of learned genetics. And when I came to Brazil and then I started working here, I think this was the most striking thing for me, because I already thought that was very complex, but then I realized if one mutation very specific, my question was first, how can people with the exact same mutation can develop such different phenotypes? And this is already something that we are doing a lot of effort to understand. And then when I came to Brazil and I started having contact more with other patients, like HCM patients, I looked and I thought, oh my God, but then you can have several types of mutations in several types of genes. This is never going to end. it has
totally different
forever, and we don't really. So we know so little about genetics, and that I think made me really put a lot of effort in cardiogenetics and be an enthusiast of showing people how important it is and to make it grow a bit more in Brazil. So yeah, more than the phenotype of the patient, which was PLN is really different than HCM. I think was really how I thought that how important genetics is and how we can actually get there. And I think coming back to the other question, the expectations you asked before, and I think this is one of my expectations working with genetics and cardiogenetics. It's actually trying to make it more common. And I think this way we can actually make panels also a bit cheaper, and we can make it more accessible and also convince, for example, public health system that is important and cost effective as well. Because once you have a patient that you identified very early, very soon, you also save money, let's say, with a lot of other therapies. So I think PLN was really my start in my cardiogenetics life, let's say. I worked only a year with PLN, so not a lot, but I think it was really interesting to see how far we can get with mutations. The point that I arrived there, we already knew where the mutation came from, how did it travel in the Netherlands, how could we find patients. So there's a lot of there's a long way to go still. So that's positive actually.
Great.
Because
Great.
I think we can do a lot of difference.
Great. Fabiano, according to your clinical practice in hasopathies, do you think that genetic tests can better clarify some overlapping features that we have in this disease? What do you think about?
Of course. So the rhizopathies is a group of diseases in the same pathway, the Has-MAP kinase pathway. So you have some conditions like Noonan syndrome that have many different genes that cause Noonan. And then you have Leopard, and you have cardiovascular cutaneous syndromes. And sometimes, many times, the same gene can cause more than one phenotype. And we had a time when the clinical examination, the morphological examination by itself could differentiate the conditions. But now we are increasingly seeing that it's not so easy to rely only on clinical diagnosis. And moreover, sometimes knowing what gene is associated can have prognosis implications also. For example, we have patients with Noonan caused by PTPN11 that rarely have cardiomyopathy. And then you have patients with RAF1 gene mutations that have a high risk of cardiomyopathy. So knowing the specific gene has important prognostic implications. And sometimes we can also have a surprise because we are increasingly seeing reverse phenotyping because in older patients, sometimes you can miss a diagnosis of Noonan syndrome, and the panel can guide you to the phenotype. genotype first is also increasingly seen on many areas of of genetics and medicine, but also in rasopathy sometimes, especially in older patients.
Yeah, and you also work with lysosomal disease, especially Fabry. Am I correct, Fabian? And what can you tell us about Fabry? And what are you working with Fabry now? No.
So I think that my interest in cardiogenetics is closely linked to Fabry disease, because Fabry disease is a condition that have different phenotypes. You have a classical phenotype in male patients that was the original description of Anderson Fabry, where you have angiokeratomas, you have a lot of different manifestations that can lead to a high clinical suspicion. But recently, they are seeing that some patients with Fabry disease have mainly cardiomyopathy, and also that sometimes it's more easy to find an undiagnosed patient with Fabry disease among a population of hypertrophic cardiomyopathy than among a population of patients with chronic kidney disease. And this motivated us to search for Fabry patients among the hypertrophic cardiomyopathy population, and also this, I think this is very, very interesting because Fabry disease is a condition that is sometimes more easy to diagnose because we have the enzyme activity that can be used to help diagnosis, but it's not so easy all the time, because specifically in those phenotypes that affect mainly the heart, you can have higher enzymatic activity and cause some confusion also. So my activity in Fabry is mainly with diagnosed patients. Most of my diagnosed patients, they have the very classical phenotype. I just have one family with the late onset phenotype, and I have a strong suspicion that many other patients with this late onset phenotypes are undiagnosed in our population.
Okay. Fernando, you also work with heart transplantation. In this scenario, sometimes you deal with that patients that have idiopathic dilated cardiomyopathy, and some people, some patients are very high risk of sudden death, and the primary prevention is sudden death in this patient. Also, always generate controversies. What do you think about the usefulness of genetic tests in this scenario?
Oh, thanks for the question, Marcelo, because this is something we've been discussing daily, especially when you are on a heart failure clinic, especially in pre-transplant or more advanced heart failure patients. You are like looking for the very sick ones, and their likelihood of having genetic mutations is higher, definitely. We are doing a study right now on sequencing these patients, taking post-transplant patients for sequencing, and also some of those with advanced heart failure. And we are finding that the prevalence of genetic mutations is higher than what is described for heart failure itself, because you have a large spectrum inside heart failure. So if you take everyone, you're going to have like 10, 20% maybe of positivity. But when you're looking into these very sick patients, you may have 30 to 40%.
Yeah.
And sometimes, and that's very interesting, you have the sum of different etiologies for those patients. So we even sequenced some patients that were ischemic, but that we know that their atherosclerotic burden or their ischemia burden wouldn't be enough for producing that phenotype.
Yeah.
So sometimes we find some pathogenic mutations. So first of all, we already found some family members harboring the same mutations. And if you're a patient with advanced heart failure pre-transplant, probably the risk of that relative is even higher than for, let's say, a regular heart failure patient. So it changes your follow-up. It changes your risk assessment. If you find a mutation on, let's say, LMNA gene or some desmosome genes that we know that are linked to a sudden death, that may change your ICD indication. And those that work with heart failure, we are always discussing, should we implant an ICD on non-ischemic heart failure patient? We have several trials saying maybe that's not the population you want to implant an ICD, but there are some mutations that are associated with sudden death. So that may change our indication. So I would say that this is a very special population. I know that not everyone is going to work with them. But if you have a young patient with heart failure or heart failure patient without an etiology and with advanced heart failure, you should definitely try to do a genetic test, because your likelihood of finding a mutation is high. And if you apply some of those criteria from Madrid that was recently published, like if you don't have a low voltage QRS on the limb voltage leads, if you don't have a left bundle branch block, if you don't have hypertension, you increase your likelihood of having a mutation. So I think you should definitely use this population for genetic testing. If you're in a scenario that you don't have tests for everyone, then this is the population for using inside heart failure, let's say.
Great. Ricardo, would you like to ask any question?
Yes. I have a question from Marta Azevedo from São Carlos, São Paulo. Sorry, I am. I was asking almost in Portuguese. Marta Azevedo is asking why you decide to be a geneticist with a likelihood to be working with cardiology and two cardiogeneticists. Why you decide to do this?
For who?
For all of them, and for you as well. But you are not so young.
In my case, I always works with cardiomyopathies, and several forms of this disease are inherited heart disease. So to study genetics, cardiovascular genetics, it's a way to have contact with the frontier of knowledge in these topics. So I think it's a natural consequence of my work, and I think it will be very common in the future only for the cardiologists, but all of the specialties, because genetics is involved with everything. So I think it was a natural consequence of my work. Fernando, what do you think about?
sometimes, Marcelo, why do you cheer for Flamengo? There's some things you cannot explain, right? Some bad choices in life, I know. But sometimes like genetics cross your path, you fell in love, and that's it. That's a simple explanation. That's things that happens in life, right? So yeah.
Yeah, I think I kind of answered this one already, but I was really actually dazzled by how big the cardiogenetics world was, because I didn't have this contact in Brazil. So before even doing the cardiology residency, I went to the Netherlands and started to have contact with it. And I was amazed by how much we can still discover and how much we can still find out about cardiogenetics. So also depends, like Fernando said, we cannot explain everything, but I think it depends a bit also on your experience in a certain group with certain people, which I can, of course, say that my PI there was Peter Van Tintelen, which is one of the speakers tomorrow. I think he was such an enthusiast of the team, and he was, how can I say, I felt so good in that group that also gave me the idea of, I liked it a lot. And once I started to understand something that was so, hard or not known by me before, I think I kind of liked it a lot and decided I wanted to do it.
Well, Fabiano's always a geneticist, so but would you like to give your opinion?
Well, as I said, I was already working with some rare diseases that affect the heart, and but we had a very limited investigation for this rare disease, and I always had the interest to go deep and allow those patients to reach their diagnosis, irrespective if it's a very rare disease or more common, like some sarcomeric conditions. I'm happy to have this opportunity right now, and I hope that our patients can continue to have access to the tests and counseling.
Ricardo, Fernando wants to talk.
Okay, Fernando, please.
No, I just want to make a question for Fabiano as well. I know it's a question about how the wolf could take care of the sheep, but I would like to hear the other ones as well. But we're talking about cardiology and genetic disease applied to cardiology, right? When should we bring the geneticists on board? Because usually we have the patient in front of us, we have the family, we order the tests, we have the results, we discuss with them. But what about the geneticists in this program? Should we always refer for geneticists for counseling? Should we send patients for geneticists only when we think there is a phenotype that we cannot diagnose and we think that patient may have a syndrome? What are your thoughts on that?
Fernando, before Fabiano answer your question, I will tell you, sometimes you must, as a cardiologist and a cardiogeneticist, send the patient for the geneticist, because there are some diseases that we are not very prone to deal with, and they are. But there are some diseases that maybe, Fernando, I will not answer for you. Please.
Well, just for my opinion about that, geneticists are rare professionals, and we are not always available to see all those patients with cardiological conditions. But I would say that ideally we would always work together, but there are some times that we can help more. And one of those situations is when we have syndromic conditions. We are trained to do the dysmorphological examination and indicate biochemical tests and evaluate for those rare diseases that our training. And when patients have some demands related to counseling, especially sometimes a patient can ask about the risk for their relatives or reproductive decisions. And sometimes I think in those cases, the geneticists can help, be very helpful, because this is an area that we are intensely trained on reproductive genetics. And I think those two situations would be the situation that we could help more. And on other situations then is helping on discussing variants and hard cases. And we are always available to help to interpret some difficult results.
Are there any red flags, like when the patient's with us, and because we are not trained to watch for syndromes? I know a case of an HCM patient that came for me. He was like short on height, and I thought, and he was young. I said, "There's something here. I don't know what it is, but there's something here." And I know that when I send you, the patient said, "Oh, of course, that's a Noonan disease," and said, "I didn't saw that coming." So because we are not like used to watch for like usually facial dysmorphologies. So what would you say that, like, for the general cardiologist, what are the red flags for syndromes that you think like we can look for and that we should be aware and then send to the geneticist?
Well, you said about Noonan syndrome, and this is a very important diagnosis on cardiogenetics. So just to use your example, if you have dysmorphic features that are not well explained, sometimes they're not familial. They're different from their relatives, or sometimes you have multiple symptoms in a single patient, like the same patient that has a heart condition also have a kidney condition, also have an eye condition, or muscular symptoms or neurological symptoms. This can guide you to a more syndromic diagnosis. And I think those would be the red flags, mainly the red flags for you to considering that maybe you have a rare disease in this case. And I think sometimes also if you detect an X-linked pattern on like mainly men are affected. This is more usual in syndromic conditions than in non-syndromic conditions, and this can also be useful to distinguish those cases.
Okay. Any other questions from the audience?
Yes, Marcelo. There is a question from a resident of cardiology from Pelotas, Lucas Ferrari. He is asking, this is a very interesting question for the young doctors, and how to be a cardiogeneticist.
I think there's no formal training for cardiogenetics yet.
No formal training at all.
So you are,
but I think, okay. Yeah, but I think there's two pathways, and they change a little bit. If you are a cardiologist and you're interested in genetics, you're gonna have one view of the things. You're probably gonna deal with more common cardiology disease, heart failure, cardiomyopathies. Of course, you have to study. You can do, let's say, a PhD on the subject, so you have a place to study, because we don't have like fellowships or residencies on that in Brazil. We do have that abroad, but that's a way of doing things, like following a clinic, academic hospital with that. You can do a PhD on genetics, but you can also follow from, as Fabiano do, from the genetic side as a geneticist, following, looking for cardiology conditions, and of course then you're going to deal with more rare diseases, Anderson Fabry, Noonan, and other rare conditions. So I think until we don't even have like a heart failure specialty recognized, so for cardiogenetic is gonna be harder. But I think these are the pathways that you can follow if you wanna look for working with this.
Thais, what do you think?
Yeah, well, that's exactly what Fernando said. There's no official training for it. So I think the best way to study it is doing an after graduation or graduation as a master or a PhD on the subject, so you can study and learn about it. I think it goes for both sides, for geneticists and cardiologists, and also, which I think is really nice, is having in the university or the service that you work, is having a board with every specialist, also bioinformatics, which brings helps a lot. So following, I think everybody can help each other out and learn about a bit about it, but there's no official training in cardiogenetics.
Okay, I think we can finish. Okay, Ricardo.
Sure.
I hope you have a productive experience, and I'm going to pass over to you, Ricardo, to close the event.
Marcelo, before closing this first night of our event, maybe a short phrase from each of them, just say something to the audience about being young doctors, starting your career, and the way you are following during this time of your career after being graduating. Thais, she's doing her PhD. Fernando finished his PhD. Fabiano finished his PhD 3 years ago. So just tell us a bye-bye message.
Well, I'll start then. I think I talked about my supervisors, which I like to look up to, and so Ricardo is also my supervisor, and I like a sentence that he has a lot, which is the patient is the family. So I really like this message to really make people enthusiastic about checking the entire family and at least sparkle some red flags for genetic diseases. So I think that would already bring a lot to our medical practice.
Well, let me do a coach talk here. Well, if you want to do something and if you want to pursue a cardiogenetic study and knowledge, the path is not going to be easy as anything in life. You're going to have to pursue it. You're going to have to work hard, make connections. But if you truly like it, well, then that's it. Then you should definitely do it. And I think that most people that I worked with and that likes genetics and works with genetics, they are usually open for talking, for teaching, and for experiencing. Let's say if you want to maybe talk to Filippo and spend some months with him, I'm sure he would welcome you. or something. So I think there are opportunities there. We don't have that many people doing cardiogenetics, so it is an open field. We're ordering more and more genetic tests, and we're gonna need people to work with. So I think that's it. Like, do it.
Fabiano?
So, to finish with my message of when you choose to do medical genetics, you use it to rare disease and rare syndromes, but it's one of the things that is interesting in medical genetics is working with other professionals and have these intersections. And I think that if you choose the path of doing medical genetics and then doing some kind of specialized area like cardiogenetics, neurogenetics, you find very good partners, and this is an excellent field to choose also.
Okay, I would like to thank Dr. Marcelo Bittencourt. He's also a young cardiologist in the field of genetics. He started this part of his work in 2016, 2017, the same time that I was beginning in this area of knowledge. And I'd like to congratulate all of you, not only for being with us in this new enterprise, but also because you will be the legacy we are doing, Marcelo, but the fruits will be with you. And I'm sure I just saw that Dr. Eliane Beuren was writing a sentence for her young doctor. And she's right. Thais, Fernando, Fabiano, they are the future of cardiogenetics. And I am more than proud to be close to these, let's say, young doctors. And tonight, they showed a lot of knowledge about the field, but more than this, they showed love. And Fernando synthesized, summarized this, and I don't know why I like, and this is beautiful, Fernando. This is beautiful. Okay, thank you very much for your participation, Marcelo. I know that you will not be with us tomorrow because you have another meeting, but tomorrow morning we'll have the cardiomyopathy part of the International Symposium of Cardiovascular Genetics and the aortopathy lecture with Dr. Arson Val. We'll have people from abroad, Dr. Peter van Tittelen from Utrecht, Holland, the Netherlands. We'll have Juan Pablo Ochoa from, he's an Argentinian guy, but he is living in Spain since 2014. Also, Martin Ortiz, another guy trained in Spain, but he is living in Rosario, Argentina. And Arsonval Lamoignier Jr., who is a cardiologist and also a geneticist, and he will be talking about aortopathies. Thank you very much. Have a great evening and enjoy the rest of the weekend. Take care and bye-bye.
Good night.
Good night.